My favorite plankter, Lingulodinium polyedra. What comes to mind when you think of the best part of going to the beach? Your answer might have been to hear the ocean waves crashing down on the shore of a sunny day.
Well,
However, on a special summer night, a wonderful glowing sight can be seen in the ocean called sea sparkle. Sea sparkle is a nickname for bioluminescent algae, which is also known as plankton, more specifically dinoflagellates. Lingulodinium polyedra is the species responsible for producing the bright blue bioluminescence in the coastal waters of Southern California, Florida, Adriatic Sea, Black Sea, and etc. They are photosynthetic, so they mainly reside in the epipelagic sunlight zone. They prefer waters at temps of 20 to 24 Celsius and salinity levels of 30 to 35%. It was originally discovered by E.A. Zernov in 1909 at the Black Sea. He noticed the rusty red stripes that were in the coast of Sevastopol, Ukraine. Lingulodinium uses chlorophyll and pyridinin, red pigment that aids in more light capture, and uses photosynthesis to turn it into chemical energy for food. This makes them photoautotrophic since they can use light energy and raw molecules of carbon to synthesize their diet. They release organic carbon that will be used up by bacteria and zooplankton. LP using photosynthesis is one of their many circadian rhythms since they take advantage of the daytime to soak in all the sun. This table shows the proteins involved in their circadian clock. Pyridinin chlorophyll protein is synthesized at a constant rate during the day for harvesting light. Luciferase, that will be discussed later on in this video, was synthesized at a constant rate during the early night. Copepods are the main enemy of this species, and they are one of the main reasons LP uses bioluminescence. Starch accumulation is a common response to nitrogen depletion. As you can see in the image, it starts off really small, and then the starch begins to take up most of the cell. The chloroplast becomes smaller and less abundant during the day to prevent damage to these cells. L. polyedrum are symbionts known as zooxanthellae by providing carbon to hosts. Examples of these hosts could be jellyfish, anemones, and all the life that is supported by this habitat that is also supported by LP. L. polyedra clings to other plankton and plants as a means of transportation. If they aren't on the Plankton Express, they are riding the currents. These wave currents can be a stimulus to Lingulodinium species if strong enough, which is why their bioluminescence can be visible without any other external factors. This is my favorite plankter since I was very fascinated with the mechanics that are involved with how they glow that bright blue color. Also, for this being a unicellular organism, it is mind-blowing how they have many detailed processes with complex features. Luciferin, a bioluminescent molecule, produces this light, which is then processed by them on their own. Luciferin is the substrate to the enzyme called luciferase. That is also found in fireflies. This reaction occurs in the synthon that will be explained by Dr. Danis.
The dinoflagella is disturbed, a membrane channel is opened that allows hydrogen ions to flow into the synthon. The pH drops, the luciferase is activated, and the electron is set into motion, climbing up an energy level before falling back down, releasing a photon and causing a flash of light.
As mentioned earlier, this flash of light is used as a defense mechanism to ward off copepods. The copepod quickly rejected the flashing cell. Since in this video the copepod did not seem startled, it appears that the bioluminescent was taken as a warning. This video would support the theory of their flashing light being used for warning coloration. I was not able to find information to back up what I noticed in this clip, but it appears Lingulodinium polyedra demonstrate a domino effect when the person was walking on them. This unicellular organism has evolved at least 40 times to develop the chemistry aspect of producing a sufficient light to help defend against predators. An example of this adaptation is the circadian clock, based on only using the bioluminescent at night and inhibiting the reaction in the day when it's not visible, as you can see in the diagram. This allows Lingulodinium to minimize unnecessary energy spent on bioluminescence and save the energy for other necessary reactions like photosynthesis during the day. LP belongs to the eukaryote domain, and their clade falls under the Alveolato. The phylum is Dinoflagellates. LP and Alexandrium make up the sister group. Lingulodinium polyedra are armored polyedral cells that lack spines. They have strong ridges, and chloroplasts are also present. A flagellum is present, which is why the species are quite excellent swimmers for plankton. The cingulum that is seen in this diagram causes a rotation that safely aids the LP to suspend in the water column. Gonyaulax polyedra was the original name but was changed to be aligned with its spiny cyst. Now its name is based on further knowledge of morphology. One experiment aimed to study the behavior of these organisms showed that in large culture beakers they formed vertical bands, and in petri dishes they formed oval swarms that were due to the difference in the light source. At night, a line of cells formed at the bottom of the petri dish that were immotile. Before dawn, the cells rose to the surface to form swarms. They then moved at high speeds. Cyst formation is a physiological response to environmental stresses. There are two types of cysts. Temporary cysts form asexually when placed under poor conditions of temperature, pH, or salinity, which causes them to stress. Formed by shredding thecal plates and flagella and forming a follicular layer and forming a more rounded shape for the cyst to emerge. Permanent cysts are formed sexually that are covered with spines. Cysts give them the ability to remain in sediment for long periods of time when placed in a hostile environment. When LP are not dealing with stressful conditions, they prefer to produce asexually, which involves binary fission. They bud off parents and develop through mitosis. If Lingulodinium are in a too perfect environment that only supports them, this can lead to some major concerns. LP play a major role in red tides, also known as harmful algal blooms, due to their high density on the surface, which creates a goldish red color. Red tides occur when they reproduce and replicate fast due to the abundance of organic nutrients from runoff. Red tides can last days, weeks, or months depending on the area, which can be May, June, or even October. They release toxins that are polyether toxins, which is highly toxic to marine life. It is not advised to go swimming even in the bioluminescent waters due to these toxins. The accumulation of toxins will be considerably high during a red tide, and it is hard to determine when this is. However, sometimes the toxin levels are low enough to not cause harm, which is why you can see clips of people surfing, swimming, and cannonballing with Lingulodinium. What are the consequences of the people that end up swimming or consuming the toxins through seafood during red tide? Since these toxins can accumulate in high concentrations in shellfish, if consumed, these toxins can lead to loss of appetite, swollen belly, nausea and vomiting, abdominal pain, and indigestion, which could lead to neurological damage or even death. If caught in proximity of this affected area, it can lead to allergy-like symptoms such as sneezing, labored breathing, rash, runny nose, edema, cough, and other symptoms. Lingulodinium polyedra are a key component to their food chain since they are photosynthetic and provide a food source to zooplankton that works its way up to filters, predators, and top predators such as large sharks. These organisms have a lot more significance in the marine environment than just being the ocean's blue light of darkness.